Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, welcome to the medical imaging session. You okay, Ryan? And this afternoon we have three speakers. Because of the late start, we're going to just be trimming the talks by 5 minutes apiece. I think we can do that. Thank you to my speakers. And first up is Emilio. And he's going to be talking, you can see the title on the screen there, Scalable and Interactive Segmentation and Visualization of Neural Processes in EM Datasets. I recognize one of the names there. Hans-Peter Pfister is one of the authors on the Fourth Paradigm book. So if you haven't got to that section in the book yet, please look out for that. Without further ado, Emilio, over to Emilio. Thank you.
Everyone, can everybody listen to me? Okay. So hello everyone, my name is Emilio. I'm a grad student at Tufts University and a researcher at the Center at the Initiative in Innovative Computing at Harvard University. The title of my talk is Scalable and Interactive Segmentation and Visualization for Neural Processes in EM Datasets. Before I start my talk, I'd like to thank Microsoft Research for their generous support with the project and for providing the funding to conduct this research that I'm presenting here today. I would also like to say that this is joint work between the institutions and centers that you can see on the bottom of the slides, Tufts University, the University of Utah, VRVIS in Vienna, and Harvard University. So most of us will probably agree that one of the most important scientific challenges in this century is understanding how the brain works. And it is widely accepted in the neuroscience community that the wiring diagram of the brain determines to a great extent how information is stored and processed in living organisms. So one of the questions that neuroscientists are asking themselves right now is, given the full brain of an animal, how can we determine its wiring diagram? So this is exactly what connectomics is all about. And one way of looking at this question is the way we do this in our lab. Let's say we have the full brain of a mouse. We can cut a small sample from the area that we are interested in analyzing, and then we can peel this sample using a very thin diamond knife, and we can stain this sample, and we can get it ready in wafers for an electron microscope. Once we have the sample in the electron microscope, we can acquire the images from the brain tissue. So the result of this process looks like this. Here we can see the cross-sections of axons and dendrites in the brain. We can distinguish myelinated axons from non-myelinated ones. And we can also see small cellular structures such as mitochondria or synaptic neurotransmitters. So this is a great technology to image the brain, but it comes at a cost. This imaging technology yields resolutions of about 3 to 5 nanometers in X and Y and 30 nanometers in Z. And this means that any meaningful research that we want to conduct using this data will most likely involve the analysis of hundreds of gigabytes. It also means that any computer vision method that we want to apply on this data, such as registration, segmentation, stitching, visualization, will be computationally expensive, or if not impossible with today's computers. So this is exactly the motivation of our project. We want to make it easier for neuroscientists to be able to analyze this data and make discoveries about the connectome of the brain. And to give you an idea of some of the tools that neuroscientists are currently using to analyze this data, this is a quick demo of Reconstruct. It's a software program that is about 3 years old and is currently used worldwide to do neuron reconstruction. The way this program works is the user is given an interface where he can outline the 2D cellular boundary of the cross-section of a neuron and then move to the next section and repeat this process, section by section. And by doing this, the neuroscientists are telling the computer what pixels should be grouped together in order to form a neuron. So, as you can imagine, this is a very labor-intensive effort. It's also very time-consuming, especially when we are dealing with thousands or millions of neurons and also of gigabytes of data. So we can summarize the motivations of our project in 3 main points. First, most common segmentation methods for neuron reconstruction rely too heavily on the user. That is, they are too manual. Also, there are not any existing 3D visualization tools that can handle this type of data sets. And also, to the best of our knowledge, there are not any existing efficient workflows that really just help the neuroscientists focus on the science. So this is why we are presenting here NeuroTrace. NeuroTrace is an integrated framework for visualization and segmentation. Sorry for the problems with the video. Using NeuroTrace, neuroscientists can choose a region of interest within the volume, and then with only a few scribbles and a click, they can follow an axon section by section until they can fully extract the neuron from the data. Also, at any point during the segmentation, the user can pause the segmentation and he can zoom in, rotate the data in real time, and see both the segmented results and the original data simultaneously on a normal computer. Best of all, again, this is something that runs on a normal computer, any Windows machine that has a GPU. So NeuroTrace is essentially an integrated solution for scalable and interactive segmentation and visualization of EM datasets. And we claim that the contributions we are making with this tool are the following. We have a tool for semi-automatic segmentation of neurons in EM datasets. It's a tool that offers integrated real-time visualization of both data and segmentations. And we are also presenting an interactive workflow for the general problem of neuron reconstruction. So this is the workflow that I'm talking about. It's the workflow that we had in mind when designing NeuroTrace. The user typically goes through this workflow from left to right, and it's made of 3 main components. In the first part of the workflow, we have the pre-visualization for the selection of the region of interest. In this part of the workflow, the user can use volume rendering to choose a region of interest from the data. And for the computer graphics people, this is the unit in charge of doing real-time raycasting. The second component of the workflow is the part in charge of the interactive segmentation of neurons. In this part of the workflow, the user can interactively segment axons in a semi-automatic manner as she flies through the data. And the last component of the workflow is neuron reconstruction. Once we have the segmentation results, the question is how do we collect this data and compress it for visualization and storage. So let me go through each of these components one by one. The first one is the pre-visualization of the data for the selection of region of interest. So one of the most important features of the pre-visualization of the data is the ability to do on-demand volume rendering of the data. And the goal here is to avoid the any pre-processing or any generation of extra information during the raycasting. So we can achieve this if we, for example, process only those data blocks that are visible to the user at any point, and also if we take advantage of GPU caching schemes so that we can quickly recall those data blocks that have been already processed in some previous interaction. So here is a demo of how this pre-visualization would work. Here you can see how the user can quickly move across sections on real time. It also shows on-the-fly edge detection and denoising of the data. And this is one of the data sets that we acquired. It has 1K by 1K pixels by 100 sections. And again, this runs on a Windows machine as long as it has a GPU. So that was the first part of the workflow, the pre-visualization of the data, so that the user can choose a region of interest. The second part is the segmentation of neural processes. So the most important thing here is that in order to extract a full neuron from the data, we first make the approximation that an axon can be decomposed as a collection of 2D rings. Now, these 2D rings are essentially the cross-sections of the axon, and they correspond to a sequence of 2D segmentations. The benefit from this approach is that we can look at the segmentation of a neuron as the tracking of its cross-sections as we fly through the data. Now, this is very useful because it's easily scalable to large data sets, because we don't have to run a full 3D segmentation method on the data if we just want to follow a neuron through hundreds of gigabytes. And also, the way this would work in practice is the user would choose a region of interest using the previous part of their workflow. Then she would draw an initial 2D boundary on any cross-section of the neuron, and the computer will automatically iterate between automatic 2D segmentation and 3D tracking. So here's a demo of how this would work in practice. You can see that starting from the ground truth of the segmentation of some cross-section of the neuron, the computer can propagate this solution and track the cross-sections automatically until it fully extracts the neuron. Again, the main strength of this is that at any point during this segmentation and tracking, we are only accessing pixels from two neighboring cross-sections, and therefore we keep the memory usage and data bandwidth at a minimum. Also, we can easily parallelize this tracing process if we can track multiple neurons simultaneously on different computational threads. So the question is, how do we do the 2D segmentation and how do we do the tracking? So for the 2D segmentation, we have a technique that we call active ribbons, which are essentially deformable models that can mimic the behavior of an elastic band. And here's a video of how this works. These deformable ribbons are essentially deformable models that can change their geometry and their topology until they can capture the desired boundaries. I'll play the demo again. Here you can see how the user can just specify roughly where the cellular boundaries are, and the computer can find them automatically. So this is the way this works is basically using dynamic force fields that push each side of the ribbon towards each other and towards the cellular boundaries. So once we have this solution from one of the cross-sections of the neuron, the question is, how do we propagate this solution through the rest of the neuron automatically? So in order to address this question, we have two steps. First, we estimate a new 3D coordinate system dynamically for each new cross-section. And for this, we have a linear filter. It's a linear dynamical system. And that gives us a new 3D reference frame on the new cross-section. And then, in order to compensate for any misalignments in the data or any non-linearities in the motion of cross-sections, we also apply nonlinear image registration for every two adjacent cross-sections using optical flow. So the way we can look at optical flow this, as another force field that is acting on our deformable ribbons, and they push the ribbon towards the registered images. The good news is that optical flow is a very spatially coherent operation, which means that can be easily optimized in a GPU. So here on the right, you can see the solution that we would get if we just use the linear filter to initialize the ribbons. And you can see that we can't enclose correctly the cellular boundaries, and this is why we need some nonlinear registration process that can push the ribbon towards the right solution. So we've seen so far how the pre-visualization and the segmentation works. The last part of the workflow is neuron reconstruction. And by reconstruction, I mean the identification and representation of the 3D region of a space that encloses and fits correctly each neuron. So as you've seen, all we have so far is just a collection of rings. So the question is, how do we efficiently collect this data and compress it for both visualization and for storage? So again, we can answer this question in two steps. First, the compression of the data. We can easily fit 3D ellipses to each of the ribbons of the cross-sections of the neurons, and we can do this on the fly as the user is segmenting the data using a GPU, and this allows us to move from a pixel-based resolution-dependent representation of the ribbon to a parametric representation where we only need 10 parameters or 10 numbers to represent each cross-section of a neuron. Once we have the compressed data, now the question is, how do we visualize this data as we are collecting it? So this is why we have something that we call on-demand single-pass raycasting. If you are familiar with raycasting, you would know that one way of visualizing a surface is by casting rays from a camera and finding where the rays intersect with the surface. Now, this is a very easy operation if we have an implicit function that tells us what's inside and what's outside an axon. But in our case, we don't have this phi function, and all that we have are just the ellipses that we interpolate from the ribbons, from the cross-sections of the axons. So we don't have phi, but that's why we can do real-time 3D ellipse interpolation, and for every ray that we cast from the camera, we can estimate what's the best ellipse that describes the axon, and from this ellipse, we can compute on the fly the phi function, and that tells us where the axon is. So here is a demo of this interactive or real-time raycasting of both the data and segmentation results. You can see that the segmentation blends very smoothly with the original data. Again, this is interactive, real-time, on a normal Windows computer with a GPU. And we can also have some nice transparency effects, which is actually really useful when the user wants to explore the data and the segmented results. So we've seen already the different parts of the workflow of NeuroTrace. Now we can move to the results and the discussion of the project. So it is clear that the GPU is what makes NeuroTrace a useful tool. The GPU allows us to do all these fancy computations on real time at interactive rates. And using NeuroTrace, the accuracy of the segmentation is about 80%. So we only require about 10 or 20% of manual edits per axon. And this means we also can we can process about 3 slices per second using the GPU on a normal computer, which is equivalent to about 1 millimeter of brain tissue per day. And we also conducted a user study with the undergrads in our lab, and we got very positive results. Here you can see the accuracy of the segmentation done by a variety of users. And you can see with Reconstruct, which is the first tool I showed you on the talk, you see that the accuracy of the segmentation varies strongly. This is with respect to the ground truth of an expert user. And with NeuroTrace, we get more consistent and, on average, better accuracy on the segmentation. So this is some of the feedback we get from the users that use NeuroTrace. NeuroTrace is a tool is a lot easier to use than other tools like Reconstruct. It's more efficient, trustworthy, and very important, it's easy to correct errors with NeuroTrace. Because if there is any error, the user doesn't have to redraw the outline for every cross-section of the neuron. She can just brush the elastic band on some cross-section, and the computer will propagate the solution to trace the neuron completely. So, in conclusion, these are the take-home messages from the talk. We have presented NeuroTrace is a GPU-accelerated framework for both segmentation and visualization of neurons in EM datasets. We can handle arbitrarily large datasets because we are only accessing those pixels that are needed for segmentation and visualization and tracing. It's intuitive to use, and it also provides interactive performance. For future work, we are already working on a GPU client-server architecture of NeuroTrace, which is going to be useful so that we can run NeuroTrace on a laptop or a lightweight computer without having a powerful GPU. And we're also trying to implement on-the-fly stitching and registration so that we can have stitching, registration, segmentation, and visualization in just one tool. Thank you very much. You have any questions?
Yeah, a couple of questions at the back. I'm going to throw the mic at them.
The alignment issue, how do you resolve if you have severe misalignment?
So prior to this, we actually have to pre-process the data. So that's one of the things that we haven't mentioned. Yes, the data has to be collected first. There's some stitching, some alignment, but we might still have some misalignment errors, and that's what we have to compensate for them during the segmentation. So all we need is just a roughly aligned stack of images. It doesn't have to be fully aligned. But yeah, some alignment has to be done before.
There's a second question at the back.
Yeah. So actually, we don't have a video of this, but we have already a version of NeuroTrace that can, the user can just give the initialization for multiple cross-sections, and then both those ribbons will run automatically, simultaneously on the same data, because they are just accessing different parts of the data sets. So there are some issues with the atomicity of the operation sometimes when two ribbons try to access the same parts of the data sets. But yeah, it's fully parallel disabled, so we can easily track multiple neurons on different threads.
I thought rather it might be useful for your precision, because if you, I guess that some of the mistakes you might get if the system makes a mistake is that the ribbon gets a bit too big. But if there was a neighboring ribbon which says, oh no, you're going into my sequence, maybe you could give some feedback there. Have you thought about things like that?
Yes, and actually, so there's one paper we have in CVPR, the Computer Vision and Pattern Recognition Conference, where we talk about repulsion forces between ribbons. So the great thing about this framework is that each of these ribbons basically have force fields. And when they get too close to each other, we can build dynamic repulsion forces between them so that they don't get into each other's pixels. Yeah, that's actually, we don't have a video of this here right now, but it's on the paper. Sorry.
Fabio, you have a question?
Oh yeah, quick question. It seems to require the interaction, an initial input from the user to specify an initial boundary.
Yeah.
How hard is to make it fully automatic?
So that's the holy grail of the project, right? How do we do this completely automatically? So we are looking into this right now. We are looking into Markov random field approaches for this problem so that we can easily just know how many neurons there are in the data set. How can we process them automatically? We don't have anything that is fully automatic yet. We are looking at that right now. But in the meantime, we have something that the neuroscientists can use to just trace individual neurons in the data. But that's the holy grail, yeah. How do we do this fully automatically?
Fabio, would you like to get set up? I had one more question myself.
Yeah.
Are you performance bottlenecked in any way at the moment? So you're using the GPU, you're saying it won't work on a regular laptop without
Yes.
GPU. I saw NVIDIA there, presumably you're using CUDA for...
Yes, we do.
Are you bottlenecked on the GPU today, or is it more disk access?
Right now, the bottleneck is actually the alignment of the data. So once we have the data aligned, it's something that we have a GPU cluster, but we are working on a solution that, we can process 1 millimeter of brain tissue a day, which is enough for some neuroscience.
Okay.
But right now, the biggest bottleneck is we have automated the collection of data, but the alignment, the pre-alignment, it's what it actually is difficult to do.
Great, thank you very much.
Thank you so much.
Speaker once again, thank you.
Thank you.